Hello, welcome to Biology, the scientific study of life. More about glycolysis harvests chemical energy by oxidizing glucose to pyruvate. The word glycolysis means splitting of sugar, and that is exactly what happens during this pathway. Glucose, a 6-carbon sugar, is split into 2 3-carbon sugars. These smaller sugars are then oxidized, and their remaining atoms rearranged to form 2 molecules of pyruvate. Pyruvate is the ionized form of pyruvic acid. As summarized in this diagram and described in detail next page also. On the next 2 pages, the pathway of glycolysis consists of 10 steps, which can be divided into 2 phases. During the energy investment phase, the cell actually spends ATP. This investment is repaid with dividend. During the energy buy-off phase, when ATP is produced by substrate level phosphorylation and NAD positive is reduced to NADH by electrons released from the oxidation of the food, glucose in this example. The net energy yield from glycolysis per glucose molecule is 2 ATP plus energy yield from glycolysis per plus, notice that, 2 plus NADH. Notice that all of the carbon originally present in glucose is accounted for in the 2 molecules of pyruvate. No CO2 is released during glycolysis. Glycolysis occurs whether or not oxygen O2 is present. However, if O2 is present, the chemical energy stored in pyruvate and NADH can be extracted by the citric acid cycle and oxidative phosphorylation respectively. Notice the closer look at glycolysis. The orientation diagram at the right traces glycolysis to the whole process of respiration. The steps also, step number 1, energy investment phase. Glucose enters, notice this, glucose enters the cell and is phosphorylated by the enzyme hexokinase, which transfers a phosphate group from ATP to the sugar. The charge of the phosphate group traps the sugar in the cell because the plasma membrane is impermeable to ions. Phosphorylation also makes glucose more chemically reactive. In this diagram, the transfer of phosphate group of electrons from one reactant to another is indicated by coupled arrows. Notice that. Stage 2, glucose 6-phosphate is rearranged to convert it to its isomer, fructose 6-phosphate. Notice that. Step 3, this enzyme transfers a phosphate group from ATP to the sugar, investing another molecule of ATP is glycolysis. So far, 2 ATP has been used with phosphate groups on its opposite ends. The sugar is now ready to be split in half. This is a key step for regulation of glycolysis. Fractokinase is allosterically regulated by ATP and its products. Step 4, this is the reaction from which glycolysis gets its name. The enzyme cleaves the sugar molecule into 2 different 3-carbon sugar, dihydroxycetone phosphate and glycyrrhydro 3-phosphate. These 2 sugars are isomers of each other. Step 5, isomers catalyzes the reversible conversion between the two 3-carbon sugars. This reaction never reaches equilibrium in the cell because the next enzyme in glycolysis uses only glycosylhydride 3-phosphate as its substrate and not dihydroxyacetone and not dihydroxyacetone phosphate. This pulls the equilibrium in the direction of glycerylhydride 3-phosphate, which is removed as fast as it forms. Thus, the net result of steps 4 and 5 is a cleavage of a 6-carbon sugar into 2 molecules of glycerylhydride 3-phosphate. Each will progress through the remaining steps of glycolysis. Step 6, energy path of phase. This enzyme catalyzes two sequential reaction while it holds glycerylhydride 3-phosphate in its active site. First, the sugar is oxidized by the transfer of electrons and H positive to NAD positive, forming NADH, a redox reaction. This reaction is very exergonic, and the enzyme uses the released energy to attach a phosphate group to the oxidized substrate, making a product of a very high potential energy. The source of the phosphate is the pool of inorganic phosphate ions that are always present in the cytosol. Notice that the coefficient 2 precedes all molecules in the energy path of phase. These steps occur after glucose is split into 2 3-carbon sugar. Step 4. Notice this one also, step 4 or step 7. Glycolysis produces some ATP by substrate level phosphorylation. The phosphate group added in the previous step is transferred to ADP in an exergonic reaction for each glucose molecule that began glycolysis. Step 7 produces 2 ATP since every product after the sugar splitting step 4 is doubled. Recall that 2 ATP were invested to get a sugar ready for splitting. This ATP depth has now been rebuilt. Glucose has been converted to 2 molecules of 3-phosphoglycerate, which is not a sugar. The carbonyl group that characterizes a sugar has been oxidized to a carbonyl group COOO minus, the hallmark of an organic acid. The sugar was oxidized in step 6, and now the energy made available by that oxidation has been used to make ATP. Step 8. Next, the enzyme regulates the remaining phosphate group. This step prepares the substrate for the next reaction. Step 9. This enzyme causes a double bond to form in the substrate by extracting a water molecule, yielding phosphonyl pyruvate (BEB). Phosphonyl pyruvate (BAB). The electrons of the substrate are rearranged in such a way that the remaining phosphate bond becomes very unstable, preparing the substrate for the next reaction. The last one, or the last reaction of glycolysis, produces more ATP by transferring the phosphate group from BAB, phosphonyl pyruvate, to ADP, a second example of substrate level phosphorylation. Since this step occurs twice for each glucose molecule, 2 ATP are produced. Overall, glycolysis has been 2 ATP in the energy investment phase, steps 1 and 3. The produced 4 ATP in the energy path of phase, steps 7 and 10, for a net gain of 2 ATP. Glycolysis has rebuilt the ATP investment, which 100% interest. Additional energy was stored by step 6 in NADH, which can be used to make ATP by oxidative phosphorylation. If oxygen is present, glucose has been broken down and oxidized to two molecules of pyruvate, the end products of the glycolytic pathway. If oxygen is present, the chemical energy in pyruvate can be extracted by the citric acid cycle. Okay, thanks for listening.